Okay, it's 8 p.m. Eastern Time. I'm Pietro, Michelucci, and I'm here with Dr. Chris Schaffer. We're at the Cornell Department of Biomedical Engineering, broadcasting live in the final hour of the Crush ALZ competition. So this is the first time we've done a live streaming event like this, and we're very excited about the reason that we're here, and that reason is that many people have dedicated a lot of their personal time to helping advance Alzheimer's research by playing stall catchers. And so I'll just start by introducing myself. So again, I'm Pietro Michelucci, and I direct the Human Computation Institute. Our flagship project is Eyes on Als, and the game Stall Catchers is part of that project. I'll go ahead and introduce Chris, and then tell you a little bit more about what I do after that. So Chris is a professor of biomedical engineering here at Cornell University, and he co-leads the Schaffer Nishimura Lab. And so Chris, could you please talk a little bit about the work that you do here, and in particular in the Alzheimer's research?
Yeah, thank you, Pietro, for the opportunity to come and talk with folks about the work we're doing. Well, the echo is really delayed.
Okay.
It's fine. So like Pietro said, I'm a faculty member here in the biomedical engineering department at Cornell University. And one of the things that my and the Zomi's lab is trying to do is to understand why brain blood flow is decreased in patients who have Alzheimer's disease. And the thing we found in mice that are genetically engineered to get this disease is that the smallest blood vessels in the brain tend to get plugged at higher rates in Alzheimer's disease as compared to normal mice. But it's hard to do this. It's hard to find those little plugs, and that's what Stall Catchers is all about.
Great. If you take those out, does that help? Maybe. I don't know if you even need them. So we call it technical difficulty. Maybe just mute your audio, and then we're good. Like I said, it's our first time doing this kind of thing. Part of what makes this project work is that we have folks like Chris and his colleagues here who are doing the challenging work, and in terms of the Alzheimer's research, and then the other piece is the Eyes on Als part of the partnership, the human computation side. And that's about building a crowdsourcing platform that allows people to play a game, and in the course of playing that game, actually help advance the research. And so there's actually a whole body of work that's evolving in building what we call citizen science platforms like this one, that allows anyone, folks who are not biomedical experts, including myself, I'm not a biomedical expert, to go online and actually contribute to the research. So it's a very fruitful partnership. And then the third tier to that partnership is you. So the folks who are actually participating in this, obviously we wouldn't be able to advance the research without your efforts. And so by playing this game, every time you annotate a vessel, you're getting us one step closer to an Alzheimer's treatment. A question that's actually come up a couple times today, and it's come up before, is people are concerned about what if they give a wrong answer, and what if they give lots of wrong answers? Are they going to mess up the research? Is it going to is their contribution worthwhile? Are they wasting their time by participating? And what we've been able to do is come up with a way of processing the information so that it's basically impossible to mess up the research. If someone were to just close their eyes and randomly pick flowing and stalled, the worst thing that could possibly happen is that you're not adding to the research. And anyone who keeps their eyes open is almost certainly contributing to the research. And that blue tube that you see, that sensitivity bar, is actually a measure of your contribution level. So as long as that bar is not empty, then that tells you how much each answer you give is contributing to the research. If there's any blue at all, you're helping and making a difference. So we're both extremely grateful. We believe this is a project that ultimately benefits humanity. It's one of these things where we're all in this together, it's great to be in this with you and working together on this. So that said, Chris, I wonder if you could just speak for a moment about the particular data set that's in the system right now, and the research question that we're in the course of answering by playing the game in this team competition.
Yeah, sure. So the question we're trying to answer right now is to determine the relative spatial location of capillaries that tend to stall and the amyloid plaques that are kind of the pathological hallmark of Alzheimer's disease. So more than 100 years ago now, Alzheimer's disease was first described by Dr. Alzheimer himself. And one of the things he noted in the brain were these clumps of very, very dense material that was just scattered throughout the brain. And Pietro, can you mute your microphone while I'm speaking?
Yeah, let me see if I can do that here quickly.
So these clumps of amyloid are, and over the years, it's turned out that these aggregates of protein in the brain have been discovered to be made of this small peptide called amyloid beta, and it looks like the accumulation and aggregation of amyloid beta in the brain is the thing that drives Alzheimer's disease. And then what we've seen in these mouse models of Alzheimer's disease is some of the capillaries have stalled blood flow. When they do, it decreases brain blood flow. And when we fix that, blood flow improves and the memory performance of the animals improves. But we want to try to understand the relationship between these two aspects of the pathology in Alzheimer's disease. Pietro, that got it. The echo's gone there. Okay. So what's happening here is we have a set of data where we've imaged the blood vessels, and through stall catchers, all of you are helping us find which of the capillaries are stalled. And then in that image, we also know where all the amyloid plaques are because we added a fluorescent dye in these animals that labeled them as well. And then once we know which capillaries are stalled and where they are, we've written software that will allow us to calculate the density or how many of those amyloid plaques there are right around the vicinity of capillaries that are stalled. And we'll compare that to the density of these amyloid plaques right around the vicinity of capillaries that are still flowing. And we're just looking to see at this point whether there's more plaques near stalled vessels, whether it's the same, or if there's even decreased plaques near stalled vessels. There's biological mechanisms that would be consistent with all three of those hypotheses. And at this point, we're just trying to understand. The goal of this is this would help us give us one more piece of evidence that could help us understand the causes, the fundamental causes of these capillary stalls. If we understand the fundamental causes of the stalls, that'll give us insights into how to develop therapeutic strategies to try to get rid of them.
Chris, can you make that a little more concrete in terms of, let's say that we discover that these stalled capillaries are actually happening near the tangles, near the amyloid plaques. Specifically, how would that help you determine, a treatment approach, versus if it turns out they're not near those tangles?
So if the amyloid plaques tend to be very, very dense right near the blood vessel, then we might be looking for a very local effect of the amyloid plaque. So it might be amyloid beta binding directly to a receptor on a cell or something like that that's triggering the inflammation. If instead it looks like the amyloid plaques are not particularly close to the places where capillaries are stalled, then we might be looking at a secondary effect of amyloid. So it might be amyloid driving an altered cellular behavior in one of the cells that sits away from the blood vessels in the brain, for example, brain microglia. And it's signaling from those that in turn is driving the vascular inflammation that causes the capillary stalls. So I don't want to mislead. Answering this question is not going to point us directly in, this pathway versus this pathway. It's something that narrows the range of options that we need to explore.
Got it. Thanks. At this point, what I'd like to do is I'd just like to mention we hope that this can be an interactive forum, and so if you're able to if you're watching the video and if you're listening to us, I would encourage you to do one of two things. Either open up your StallCatcher's browser and annotate as many vessels as you possibly can in the last hour as we push the envelope on this competition, or take this opportunity to ask us questions or let us know what you think. And we're going to be monitoring the comments that are made on the video, and hope to have a chance to answer some of those. So the next thing I want to do is just talk a little bit about the competition itself. This has really been an incredible journey for all of us. If you've been part of the competition either from the beginning, maybe you saw the PBS documentary, The Crowd in the Cloud, then you know some of the history of the project and the purpose of the project from that. And it was in the live after show social media event that we actually launched this first team competition, which was the first time that we actually opened up the ability to create teams in StallCatchers, which was kind of an experiment. So whether you were with us from day one or whether you joined recently, you've probably gotten a sense of kind of a level of excitement around the competition. There have been some very close races near the top of the leaderboard, and there have been some closer races in other parts of the leaderboard, and each day has brought one or another kind of new surprise. So I thought it'd be nice to just kind of highlight some of the interesting aspects of the competition over the last month. I do want to start by saying we're also going to monitor live what's happening in this last hour of the competition. At a very basic level, we're looking at the number of real vessel annotations that have been made just today. And I'll have to look back through our records here. But I think we've already crushed a new record today. So yeah, it looks right. So our previous record yesterday was that over 23,000 real vessels were annotated. When I say real, as you probably know, some of the vessels you see are calibration vessels. Those are examples where we already know the answer, and we're using those as a way to monitor your sensitivity, your ability to tell which ones are flowing and stalled, and as a way to give you opportunities to learn how to tell the difference between those two. So those are not part of the data set, but they're a necessary part of the project. The rest of the vessels, the ones where we say maybe, the ones where they're kind of disappointing because you see the maybe and you don't see a lot of points, and you're like, I wish that had been one of the big point ones. I feel the same way when I'm playing. But actually, those are the ones that are advancing the research. So those are super important, and those are the ones we're counting right now. So yesterday we crushed all previous records with over 23,000 real vessels annotated. Today, Saturday, which characteristically is a slower day on stall catchers, right before we began this, I did a quick check, and we'd already annotated over 35,000 wheel vessels. So this is
amazing.
Yeah, I mean, this is obviously unprecedented, definitionally, and so over the course of the next 45 minutes, we'll check back and see how that number is changing and how many vessels are being annotated just in this hour. In fact, I will go ahead and tell you right now how many vessels were annotated just in the last 15 minutes. 175. So maybe that means you're listening to us instead of annotating vessels.
Or it's dinner time.
Right. Yeah. Right. Or I wrote my query wrong. We'll hope it's the former, not the latter. So as far as highlights, and Chris, I don't know, have you participated in the competition? Have you annotated some vessels along the way?
I have. I was very proud that the very first day of the competition, I was at the front of the leaderboard for at least a part of the day. I think that's in part because I had insider knowledge about when it was going to start and those kinds of things, and I've been completely unable to keep up with the level of commitment that we see from folks around the world who are playing stall catchers. It's actually truly humbling to have people devoting this much time and effort and energy toward the research that we think will really have will potentially make a big difference in Alzheimer's disease.
So on this, just as a reminder, during this last 24-hour period, we are doubling all points on stall catchers. If you've been playing, you probably noticed that you're suddenly getting much higher point numbers for each vessel that you've annotated. I would like to honor our most active stall catchers from this day. So you may have noticed that the reason for the double points is not only that it's the final day, but we're also celebrating Citizen Science Day. And so what is Citizen Science Day? I mentioned a few minutes ago that citizen science is kind of a general term to describe projects like these, where you have participants from the general public contributing to the advancement of science. And there are actually many projects around the world that are raising awareness and including more people in citizen science activities of all kinds. So on this day that we're doubling points and celebrating Citizen Science Day, I just want to honor our top 10 contributors today. Our very top contributor, I don't know if I'm going to say this right, username JNAX with an X at the end. And so I'll just mention that 1, 2, 3, 4, 5, 6, 7, 8 of our top 10 contributors today are from middle school STEM. And I will talk a little more about middle school STEM, which is a very special team. So that's our highest contributor, JNAX, with over 11.5 million points. Then we have WCHRI 7821 with 11.5 million. Aiden's Catching Stalls at 3.8 million. G. Calkins with Stall Destroyers team with 3.2 million. A. Brow 7488 with 3 million for middle school STEM. And by the way, the first three I mentioned were also from middle school STEM. Aiden Horner, 2.87 million. A. Davis 5, 2.67 million. Flareon Lover with 2 million for middle. And I'll just make a comment about Flareon Lover. I happen to notice the Pokemon reference because my son is at a Pokemon tournament, a regional competition this evening, and he would appreciate that the Pokemon Flareon is mentioned in this top 10 leaderboard. So that's for my son that I mentioned that. Then we have P13490, also for middle school STEM with 1.8 million. and user Capra-Ram with 1.7 million. Glad to see him on the leaderboard. He's been, one of our highest, most active contributors since near the very start, possibly the very beginning of stall catchers. So you'll certainly see him on the individual leaderboard. Okay, so I just got a note from my colleague Agla Ramanauskaita, who you might recognize her name. She's the one who's developing the wonderful blog posts that hopefully you're reading every day and keeping the excitement alive with this competition. And Agla says that we have, let's see, already 152,000 points since this hangout started. So it sounds like we are still moving forward at a good clip. Let's see. Wonder if I don't know if this Angela is going to let me know if there are any new questions popping up, or maybe we need to set it up. Hopefully, people are able to post to this. So I did mention recapping part of the competition, and Chris, as you probably recall, I know Nozomi was actually the one on the video at that time with that hangout, but I imagine you were, well, actually you were on the other side of the continent, weren't you?
I was on a plane.
Oh, you were on a plane, so maybe you didn't see what happened, but during the first hour of the competition, 1,500 vessels were analyzed, which was like what we would typically see just in a single day of stall catchers. And then that climbed to 3,000 over the next 3 hours. Can you say something about how long it would take to analyze that volume of vessels, just a lab technician in your lab?
Yeah, so so for us, we in a single imaging session will visualize about 1,000 vessels, and it takes about 2 hours for someone who knows how to run our microscopes and things like that to do all the work necessary to collect that data set. To annotate individual vessels as flowing or stalled by a single individual in my lab who's done a lot of that would take more than 40 hours of work to analyze those 1,000 vessels. So this is basically doing the work that would take our lab, one person in our lab, a full week of doing that and nothing else, and getting it done in an hour. It's an amazing speed up of the pace of and the tempo of our research.
That's tremendous.
And we are now planning studies entirely around the capability of stall catchers. So we are collecting data now that we know that if we didn't have stall catchers, we would not be able to analyze it in a reasonable amount of time. In fact, we wouldn't even be doing these experiments because the analysis would be so hard. We would be focusing on a narrower set of questions. We wouldn't be taking the sort of risky questions and things like that we're doing now, because we would be concerned that we would put a lot of analysis into it and it would be a dead end. Now, that means we would be asking and answering more conservative questions, which inevitably leads to a slower pace of research. Here, we can ask not only those conservative questions, but also a broad range of riskier questions, which, if they turn out to be right, will give us a bigger jump forward in the research. And the freedom to be able to do that is really what Stall Catchers has helped in our lab.
So I wonder if you could sort of speak to, kind of the end goal. And maybe end goal isn't the right word, because I imagine with a disease like Alzheimer's, even if you come up with an effective treatment, you're still improving that treatment. You're reducing side effects. You're looking for earlier diagnosis, different intervention points, and maybe depending on whether the person has the genetic form of the disease or the spontaneous form. Does having this additional flexibility to ask more of these research questions help you address these different aspects of what the goal is?
Yeah, in fact, maybe what I could do is I could talk a little bit about sort of three of the broad directions of research that we see going forward that are going to take advantage of the stall catchers pipeline. So the first is a series of studies that we actually just received funding from the Bright Focus Foundation for. The Bright Focus Foundation has also supported the development and deployment of stall catchers. But in this study, our goal will be to look at a series of drugs that have already been developed and shown to be safe in humans, that were designed to interfere with the ability of white blood cells to adhere to blood vessels or to follow the signals that tell them to go to places of inflammation. These drugs were developed mostly to treat multiple sclerosis, to try to improve patient outcomes after stroke by reducing inflammation. They've not been tested in Alzheimer's disease. But because they are designed to interfere with white blood cell adhesion, there's we think a good likelihood that some of them will effectively block this capillary stalling phenomenon that we've found in Alzheimer's disease. If that were true, we would then have a drug that had already been shown to be safe in humans, and which in mice we think blocks this Alzheimer's disease. And so that would set the stage for a relatively, a clinical trial in Alzheimer patients in a relatively short period of time. So the study that we'll do here is we'll take a suite of these compounds that we think are likely to interfere with stalls, and we'll give them to Alzheimer's disease mice, take the image data, and look before and after we've given the drug, send that data to stall catchers and ask whether or not these compounds are reducing stalls. And again, the ones that look like they're effectively reducing stalls at doses that would be reasonable to give to humans, that data that we collect is then what would support an application to the Food and Drug Administration and to institutional review boards in order to be able to initiate initially a small scale clinical trial in humans, where the goal would be just to see whether or not taking these drugs led to an improvement in blood flow in the brain. If that was true in humans, that would tell us that the same mechanisms that we've identified in mice causing decreased blood flow in Alzheimer's disease are also causing decreased blood flow in the brain of human patients with Alzheimer's disease.
So that all sounds very reasonable and promising. But if I were a person who either had a diagnosis of Alzheimer's disease or found a member with Alzheimer's disease, the question I would be asking is, that sounds like a lot of stuff that needs to happen. So, let's say that, let's not say best case and let's not say worst case. Let's say, reasonably with stall catchers and this ability to speed up the pace of research, and given the findings that you've had so far, what kind of a timeline would we potentially be looking at to get to an effective treatment?
So all the way to an effective treatment is very hard to predict. But what I can say is getting to the point where we would have screened, say, 15 or 20 of these compounds that we think are likely to work, we'll be able to do that within a couple of years using stall catchers. If we hadn't had stall catchers, we would have had to pick one of those and focused our efforts just on that. So that's what it's helped us do. It's helped us, instead of just picking one and really hoping that we're right, we're now able to pick a broader set of molecules to try initially, and to evaluate in this preclinical kind of research as candidates that could go forward in a small scale clinical trial.
So it sounds like you're saying, sorry to interrupt, but it sounds like you're saying that not only is it speeding the pace of the research in terms of the ability to identify one of these,
it's expanding the scope.
But it's expanding the scope. So
right.
You're not putting all your eggs in one basket.
Exactly.
As it were.
Yeah. So that's one direction of research, trying to test molecules that we think that have already proven to be safe in humans, and we think will interfere with white blood cell adhesion, to develop the data that would be necessary to support, to establish that it's worthwhile and safe to try a clinical trial in humans. But to tell you the truth, I'm concerned that blocking leukocyte adhesion, blocking the adhesion of white blood cells in your entire body, may have a suite of side effects that are undesirable even for the treatment of Alzheimer's disease. That's going to compromise an aspect of your immune and inflammatory system if you're taking these drugs chronically and all the time. I might be wrong, and there might be a dosage of those drugs that is acceptable, that reduces stalls enough, improves blood flow enough, that it helps Alzheimer's disease, that leaves enough of the immune and inflammatory function intact that it's okay. But we're not going to know that until we do trials in humans. And so I don't want to leave that to chance. So another major aspect of the work is trying to identify another way to interfere with these stalls that's more specific to both the brain and to Alzheimer's disease than just blocking adhesion of white blood cells everywhere. So that's another series of studies where stall catchers is essential. So the way this works is we're taking mice and we're injecting molecules that block different molecular little machines along a pathway that we think might be what links the amyloid beta aggregates to the increased inflammation in the vasculature that causes white blood cells to stick. And there's some of those molecular machines, those molecular interactions along that pathway, where there's drugs or compounds or antibodies or things like that we can give to the mouse that'll interfere and block that pathway. And the question is if we block that pathway, do the stalls go down? If so, that implicates that step in the pathway. So we're really trying to chase and see if we really do understand how important this pathway, actually one identified by our collaborator, Constantino Iadacola, down at the Weill Cornell Medical College. We're trying to evaluate whether or not this pathway is responsible for the capillary stalls and what fraction of the capillary stalls is responsible for. So it's rare in biology that there's sort of one pathway that does everything. It's usually multiple different parallel pathways. So, but the goal of those studies, so we're trying to not identify a specific drug that would work. We're trying to identify the place that you should build a drug to target. So this is called, target identification, or trying to identify the mechanism or the place where you want to have a molecule to interfere. That's a longer drug development pipeline, but on the other hand, one that our goal would be to try to develop brain and Alzheimer's disease specific targets, which could reduce the potential side effects that we're worried about in terms of blocking leukocyte adhesion system-wide.
So let me see if I understood all this. It's a lot of information, and it makes a pretty interesting story, I think. So there's this idea that if we can get rid of these stalls, right, then we restore the blood flow to the brain. And if when the stalls are there and you have a 30% reduction in blood flow, it would be hard to think straight, frankly.
Correct.
And so if I remember from what you said, that you have drugs that you've tried in mice that get rid of the stalls. The problem is that that's not sustainable, that it would compromise the immune system of the mouse or eventually the human if it were the same approach. And so, sure, they would have a clear brain, but they would have no immune system. And that's not really a treatment that could be a long-term.
That's the concern, yes.
And I think what you were suggesting is that maybe some amount of that if you could, not an all or nothing, you get rid of some of the stalls and you only compromise part of the immune system, then you have an improvement in the Alzheimer's and the patient remains healthy. But then the next part of your story, which sounds compelling, and maybe this is where stall catchers makes a big difference, is that if you can conduct enough different experiments, then instead of sort of treating the symptom of the inflammation, which is these stalls, you can treat a very specific process in the brain that causes the inflammation in the first place.
Exactly.
And so if we can run enough experiments and we can figure out exactly how that works, then we can shut that part off, get rid of all the stalls that are clogging up the brain.
That's right.
And keep the rest of the body healthy.
That's right.
That's the goal.
That's the goal.
Okay.
Yeah.
Yeah. So.
And then, if you like, there's one more direction where stall catchers is essential, and this is more trying to understand what role this capillary stalling might play in driving Alzheimer's disease. And the idea here is to try to understand. So we know many things that increase the likelihood of developing Alzheimer's disease. So having high blood pressure, obesity, type 2 diabetes, high cholesterol. There's a number of these sort of factors, other things, head trauma, that tend to increase the risk of getting Alzheimer's disease. But you'll notice that many of those things that I listed are fundamentally cardiovascular kinds of diseases. They're, you know, like high blood pressure or high cholesterol in your blood. Those are cardiovascular diseases. And what isn't understood very well is why is it that problems with your cardiovascular system lead to increased incidence of Alzheimer's disease, which is a problem with your brain. And now this is very speculative. This is early stage research now, but our hypothesis or our idea is that maybe these other things that are wrong with your cardiovascular system are causing vascular inflammation themselves. And in fact, there's quite a bit of evidence that supports that in obesity and with hypertension, high blood pressure, with high cholesterol, that there's increased expression of some of these inflammatory proteins on blood vessel walls. So then if you have a process that's driving vascular inflammation because of a cardiovascular problem, maybe that's increasing stalls a little bit. And then we know from recent research that if the incidence of stalls goes up and blood flow drops, that actually decreases the rate that amyloid beta can be cleared from the brain. And if amyloid beta isn't cleared from the brain as quickly, then the concentration of it in the brain goes up. And when the concentration goes up, you're more likely to get these aggregates forming. And it's the aggregates that cause the inflammation that makes the stalls worse. And it's the aggregates that cause direct effects on neurons that impact their function. So there's sort of a potential for a positive feedback cascade here where systemic vascular effects, systemic vascular disease drives inflammation in the blood vessels, which causes stalls, which decreases clearance of amyloid beta, which causes more inflammation, which causes more stalls.
So it sounds like you're describing a vicious circle, basically.
A vicious circle, and one that we can test whether or not it's there by taking Alzheimer's disease mice and giving them some of these other cardiovascular problems. So driving high blood pressure in them, feeding them high-fat diets that lead to obesity, crossing them with mice that are genetically engineered to get diabetes. Like there's a number of ways that we can take the Alzheimer's disease mice and insert these other things that are more common in humans and evaluate whether or not that exacerbates the rate of capillary stalling. And most importantly, whether our therapies that aim at reducing stalling then get rid of the synergistic effect between these two conditions. If that were true, then this would basically this would identify, and if this turn out to be true in humans, that would then identify a cohort of patients for whom you would want to start any therapy that blocked this capillary stalling phenomena long before they developed Alzheimer's disease. So people who had hypertension, people who were overweight, folks that had diabetes or other conditions that it looks like it elevates this capillary stalling, you might want to start treating them long before they develop the cognitive symptoms of Alzheimer's disease. But first we need to know if that is in fact the link, because if it's not the link, then you're just taking a drug that's not going to help you. So stall catchers is essential for those studies. We recently estimated that we would need to annotate, I think it was, what did I tell you, 250,000 capillaries in order to answer those questions across hypertension, high blood pressure, high cholesterol, diabetes, and obesity. So it's an enormous undertaking to try to sort that out. And again, one that we wouldn't even contemplate doing without the assistance of stall catchers and all the citizen scientists who help us.
Now, just to make a little sense out of those numbers, we have to take the numbers that you just gave and multiply them by 20 today.
That's right.
So we're actually talking, and that's because it takes more than one person annotating a vessel to get a crowd answer. And today, and we're developing methods to try to improve this, but today it takes about 20 answers that we combine to get one final answer. So 20 times 250,000, do the math, but we're talking about millions of vessels. And that's why we need the help. Let's, I'm going to retreat out of the laboratory for a minute. And as you can tell from listening to Chris, one of the great things about, having Chris as a collaborator on this project is, the guy and all of his colleagues are absolutely brilliant and they clearly know their subject matter. there are times I understand him more, times I understand him less, but overall, I feel like, you know, the bioscience side of this is in really good hands, and that lets me focus on the crowd science part.
And that's been I would say that's been a mutual kind of respect. It's been enormously helpful to basically have this tool be developed in a way where it's working so robustly and has been so effective at engaging all of the folks out in the community to help us with this research. So it's been an ideal collaboration.
So so let's talk about the competition for a minute. I started to talk about the recap of the past month, and I never actually did it. So why don't I do that right now? But before I do that, I'm just going to take a quick peek. And at the beginning, I told you how many real vessels have been annotated today. And just as a quick reminder, let's see, that number was 35,707. And since we've been talking here, that number is now up to 36,211. So
that's great.
Yeah, so I mean, we've already annotated a few hundred more vessels just in this short time. I think it's not at the same pace as earlier today, and I hope that means that either you're out having a wonderful evening on a Saturday night, or that you're engaged with this hangout. So now I'm going to actually recap. So we were saying that during that first hour of the competition when we launched, I think there were 3,000 vessels annotated, actually 4,500 vessels annotated in the first 4 hours of the competition. And then the very first day of the competition, we broke all previous records with 13,000 total vessels annotated. By the end of week 3, the catcher started to gain speed, and then before long, we were actually contributing up to a week of lab equivalent time per day. So, you know, when you can start to talk in those terms, that gives you a real sense of how much the research is being accelerated. You know, we're doing 1 week's worth of data analysis every day because of the work that you're doing. So that was very, very exciting to be able to get to that point. In week 4, things just started to, it was like a snowball effect. We were seeing new records getting crushed almost every day. On day 22, the total vessels annotated in 1 day, that record was broken with 16,600 vessels annotated. So, I mean, we're talking about only a week ago, that was a record. And if you think about the numbers I just mentioned, we've more than doubled that. So it's really exciting. Part of that, I think, is you know, the competition was heating up, especially in the upper ranks. We were getting some very neck and neck. In 1 day in particular, we had the top 2 teams both had points in the millions, and yet they were separated only by about 1,000. That makes me think that they're watching each other.
Yeah.
And they're saying, I'm just going to annotate enough vessels to get past the next guy. And that was just kind of going back and forth like this. So that was very exciting day to watch. And we had the real vessel record was broken on day 25 with 9,000 real vessels annotated. And then again on day 26 with almost 11,000 real vessels annotated. And then yesterday on day 29, all records were broken. We had a huge spike, which you may have seen in Egla's blog post, where we had 23,600 real vessels and almost 42,000 total vessels annotated in a single day. And at the very end of this hour, we'll have the numbers in from today, and I can tell you already that I think we're going to be breaking even yesterday's records. I don't want to speak too soon, but I think, you know, actually based on what we've already seen about the number of real vessels annotated, I think we actually have done that. Mm-hmm. So that's just tremendously exciting. Okay, so let's maybe take a minute to talk about the teams. We got about actually only 15 minutes left here.
And I probably want to take maybe 5 minutes at some point to talk about what we would envision as the next steps with this particular question, and when we can expect to have an answer.
That would be great. Yeah. I think that's a great way. So we'll save a few minutes near the end for that. So we've got about 25 teams in stall catchers. Actually, maybe it's more than that. Far and away, the biggest team, as you probably figured out by now, is middle school STEM. So this team is a middle school in let's see, it's Lake Hazel Middle School, and that's in Southwest Idaho, here in the United States. And they actually heard about the competition on the Crowd and the Cloud, documentary. And it happened to fit well with their current lesson curricula. And then one of the teachers there, Ms. Davis, set up a team, invited her students, and the word just spread through the school. And now they have over 200 players. In fact, yesterday they added 35 new players. A couple fun facts about middle school STEM. So they happen to be at the top of the leaderboard. When it's a crowd-based platform and the size of your team makes a difference, then it definitely helps to have the biggest team. They had a competition to design their team logo. If you haven't seen it yet, please check it out. I think it's a pretty clever design. And so the number of the art students in the school competed, and the winning design became the team logo. It sounds like citizen science is kind of a new concept for the students there. But because of this competition and this experience, they're actually talking about starting a citizen science club next year.
That's wonderful.
As a result, yeah. So obviously that's the kind of thing that we like to hear about. So what I'd like to do at this moment is mention that we're going to be having another hangout on Wednesday at exactly the same time, 8 p.m. Eastern time. Except this will be hosted by the Crowd and the Cloud documentary folks. So if you haven't seen the documentary itself, go to crowdandcloud.org and you can stream, the 4-part documentary series. Each part is 1 hour. The first episode of the series has a 15-minute segment about this project. And the series was created by Jeffrey Hainstyles, who, if you've heard of Cosmos, there's the more recent version of Cosmos and the original Cosmos PBS series from the 1980s with Carl Sagan. And Jeffrey was actually one of the senior producers on that series. So we're really honored to have this project featured in the Crowd and the Cloud series. And Jeffrey Hainstyles and his team will actually be hosting this hangout on Wednesday. And Ms. Davis from the middle school STEM team will actually be joining on that panel, and we'll get to hear from her as well.
That'll be great.
About what that experience is like. So I'm really looking forward to that personally. So I spent a lot of time talking about middle school STEM, and I think there's so much more that we could say about that. But I do want to mention, because there are a lot of other teams. Not all of them are huge teams, but many of them have made a really huge impact. And again, every single person who plays, if one person annotates one blood vessel, they've made a contribution to advancing this disease. We have, 7 billion people on the planet. If each person annotated one vessel, we'd be done. So it's kind of the way I do the math. So KMS Genius Hour, the second largest team with 54 participants. Alzheimer's Research UK. These folks are also technical collaborators of ours. They're part of our scientific ecosystem. They have their own citizen science game, Sea Hero Quest, which so unlike this game where you're actually doing the research analysis, in Sea Hero Quest, you're actually being studied. So know that going in. But what they're doing is they're collecting a tremendous amount of baseline data to say, what does the distribution of normal cognitive behavior look like, so that when we start to look at people with the disease, how, can we make comparisons to those people?
Mhm.
And it looks, it looks like a fun game. So, you can check that out as well. Eyes on Owls is a team that I actually started with just trying to create a home for our own collaborators here, for Chris sitting next to me, although he went rogue on me and created the Cornell University team.
Oh, Cornell.
And there was a lot of factioning going on. I'm not going to get into the politics of it, but we're still all good friends afterwards. No, I'm just joking, but one of the great things about the Eyes on Owls team is I kind of, in the team description, I said, this is for folks who have contributed to the project. If you're not sure, whether or not you should be on this team, then you probably should. And we got a lot of folks who were also folks who participated in the early days and who have on the participant side and made tremendous contribution. And it was a real honor to have those folks on the team as well.
Agreed.
So I'm very pleased about that. And because of those people, not because of my contributions, probably Eyes on Owls is one of the biggest teams and also on the leaderboard. Cornell University is the 5th largest team with 15 people, tied with Stalls Will Not Pass, also with 15 members. Then Stall Destroyers, iWires, both with 11 and 10 players respectively. And then beyond that, each team had fewer than 10, but some of those smaller teams actually made the biggest impact. And I think Sit-Side Gamers and Citizen Science Plus were among those smaller teams where there were just some very, very dedicated players. I'm not going to list through every single team, but we're grateful for every single team that was formed, and all the statistics are right on there. You can go and look on the site. So it looks like we got about 10 minutes left. Why don't we, at this point, I'm going to take another peek. We're very close to the official end of the team competition. So I think it probably wouldn't hurt to just peek at the leaderboard right now and see what we've got. So you can look on with me here. So actually what we're seeing on this leaderboard is actually just what's happened in the last hour. So I'll refresh this. And it looks like middle school STEM is still cranking away. Even in this very final hour of the competition, they're in first place. Sit-Side Gamers, one of our ongoing leaders, also still near the top in second place just for the day. Eyes on Owls in third, Cornell University in fourth. Still that same order. And Chris, I don't know what to tell you about that. Stall Destroyers in fifth. And then if we switch to the full competition view, wow. So, I mean, these numbers have just changed so much in the past couple of days that I'm struggling to even fathom the number of digits I'm looking at. So middle school STEM currently has 90 million points. I think just 2 or 3 days ago, they were down around 20 or 30 million points. So, and last week, it was middle school STEM, Stall Destroyers, and Sit-Side Gamers, neck and neck. And pretty much through the whole competition, they've been neck and neck. But middle school STEM just took off. I can't wait to talk with Ms. Davis on Wednesday to find out what happened in the last 2 days to cause that such a huge advance.
And to Ms. Davis and all of her very dedicated students, thank you.
Also from me. So after that, we go from the major leagues here to the minor leagues, but we still have pretty close competition between Eyes on Owls and the iWires, at least for, the Eyes on Owls has 8.2 million, the iWires with 7.3 million on the leaderboard. I'm not sure that's going to change much in the next 5 minutes. But we really appreciate our friends at iWire participating in this, and we have a close relationship with iWire in that they were part of our initial team in developing this project. And this project really stands on the shoulders of two citizen science giants, the Stardust at Home project. This platform was actually built on Stardust at Home, which was a citizen science project, if you can imagine, that was created over 10 years ago. One of the pioneering citizen science projects at Berkeley University. And Andrew Westfall, the PI on that project, is a close and great member of our team. And then Sebastian Sung and Amy Robinson with iWire have been materially helpful in a lot of the, both on the community development side and on some of our data method side. So I just want to call out those. And as Chris mentioned earlier, this project wouldn't exist without Bright Focus Foundation. And we're so grateful to be partnering with them because they're not just a funding agency. They really care about the disease. They're taking the risk on, this is non-traditional research today. And but they've been risk takers, and I think because they have the history of funding the research for the first artificial heart, seeing that work out, I'm actually a personal beneficiary of some of the technology that came from that, and so grateful to Bright Focus for that. And now they're taking a chance on this project, and I think with your help, we're proving that this is something worthwhile. So stalls will not pass. Number 6 on the leaderboard. Number 7, close behind, is Alzheimer's Research UK that I mentioned earlier with the Sequest Hero game. Then Cornell University, KMS Genius Hour, and Citizen Science Plus, and that rounds out the all-time leaderboard. We've got about 3 minutes left. So yeah, why don't we take just a few minutes, to talk about, what comes next after this data set and what the answer to this question is going to lead to. And then after that, I'll give out the final numbers and declare the final winner.
Great.
We'll call it a night.
So again, the goal of this push in data analysis in this competition is to determine whether or not capillaries that tend to be stalled have a higher density of amyloid aggregates near them as compared to capillaries that tend not to be stalled. So once we know which capillaries are stalled and not throughout the vascular network, and the work you've done over the last month is enough that is going to be answered. So there's,
can I interrupt? Sorry,
of course.
1 second. We have some breaking news. One of the teams wanted to make sure that we realized that they just appeared on the leaderboard. Stalls will not pass is now on the leaderboard. Okay, please,
okay,
so
good.
Please resume.
So there's now enough annotations for us to know which capillaries are stalled and not. So it'll take us a week or two to compile all your annotations together, and then we have software that will go and look around each stalled capillary and each unstalled capillary and calculate the local density of the amyloid deposits there. And that software actually will, on this large of a data set, it's going to take it a week or two just running in the background before those calculations are done. And then it'll take us a couple of weeks of digging through that data by hand and looking at it and trying to get our head around it. But I think I would say by a month from now, by June 6th, we'll be able to post on Eyes on Owls the answer to this question, whether or not capillaries that tend to be stalled have higher densities of amyloid deposits around them or not.
I mean, to me, that it's pretty amazing that we can have that kind of a turnaround, that you can participate in a project like this. And sometimes you participate in a project, you fill out a research survey, you participate in a clinical trial, and you might hear about the results or you might not. It might be a year later. And here, and this is, I think, very important to us because you're our partners in this as well. And so we feel a sense of we need to close the loop with you about the research that's going on. And so to be able to close the loop that quickly That's always in the back of my mind. And, I think Chris cares about that a lot. And so he doesn't mind when I pester him about, new data sets and when are we going to have an answer and that kind of thing. He's always very quick to answer that and help out. And so we'll talk more on this Wednesday. I just want to, reiterate that this coming Wednesday at 8 p.m., it won't be on this Facebook page. It'll be on the Crowd and Cloud Facebook page. If you go to blog.eyesonals.com, Agla's posted all the relevant information so you know where to go to hear about that. And speaking of Agla, who's working in the background on this right now, she just sent me the final numbers. It looks like it's 9 p.m. Eastern time. This competition, let me rephrase that. This competition is officially over. We are planning more competitions, and we've got lots of cool features in store. I should probably, even though our time is sort of up here, I'll just spend 1 or 2 minutes to tell you about a feature called Redeem that's going to make all those maybes turn into gold in a couple of days. So let's look at the final numbers. If I can find them here. Bear with me 1 second here. Well, while I am waiting for those to come in, I'm going to run this query one more time and we'll get the final count. So the answer to the question about did we break a new record today, as we kind of knew the answer to that question, but now I can give you the final number is 36,318 real vessels annotated in the final day of this competition, which absolutely crushed the previous record. And can you just do the simple math and tell me what's the research equivalent of 36,318 vessels annotated?
That's 36 mice worth of data. Well, we need the 20 times over on that, though, right?
Well, yeah, that's right. So the way that we combine the answers now, you have to take those numbers and you divide by 20. The good news is, and I'll talk more about this on Wednesday, is we're improving our methods so that we can actually extract more information out of each one of your annotations. What that means is that all the annotating you've been doing, we'll be able to actually get about 5 times as much information out of that, probably in the next couple of weeks. I don't know, 5 times, between 3 and 5 times as much information out of that. So based on today's numbers, it's 35,000 divided by 20. Based on, the new numbers, it could be divided by 5.
Mm-hmm.
In that case, we'd be talking about, maybe a month and a half's worth of annotation in a single day.
Well, even requiring 20 annotations in order to have a reliable answer for each capillary, doing 35,000 in 1 day means basically doing about 2 mice. And we take data on about 2 mice a day. So this is keeping pace with the rate that we would be able to generate data from the lab, even with that.
So I've got the final standings here for the top 10. So here it is. The Crush ALZ competition ends with middle school STEM in first place with 90,937,497 points. Stall Destroyers in second place with 23.3 million. Sit-Side Gamers a close third with 21 million points. Then Eyes on Owls with 8.2 million. iWires with almost 7.4 million. Stalls Will Not Pass, 4.9 million. Alzheimer's Research UK, 4 million. Cornell University, 1.6 million. wow, that was a close. So Cornell University in 8th place, KMS Genius Hour in 9th place, also with 16 million. It looks like they're only 3,000 points apart. For a very high sensitivity annotator, that's like one vessel difference. So that was very, very close.
Go Cornell.
Go Cornell. But thank you, KMS Genius Hour, for pushing it. And Citizen Science Plus in 10th place with 1.1 million. These are significant numbers. They represent a lot of hard work, a lot of dedication, a lot of fierce competition, and a lot of people who really want to crush Alzheimer's disease. So again, we're so grateful, for your participation in this. We hope that you'll join us on Wednesday at 8 p.m. where you'll get to meet some key players, team managers, different folks on the project team who have helped to develop this project, and folks who are actually dealing with the disease on a daily basis. So that'll be, I think, a very interesting and powerful conversation to have with a lot of great people. And thanks again for participating in this competition. I did mention I was going to say something about the redeem feature, so I'll say it very quickly. All those maybes, you probably see that little redeem button there, and it says you have vessels pending. When we get final answers to whether those vessels are flowing or stalled, if all the answers you gave, whichever vessels that you annotated, you gave an answer that matches our final answer, you will get full credit, the full amount of points that you would have gotten if they had been calibration vessels. So if you've done a lot of annotating, then you're suddenly going to have a huge jump in the number of points you had. And once we get this new system going, then you'll be able to redeem almost on a daily basis. So that's something to look forward to. We're going to have eventually, hopefully sooner rather than later, an in-screen chat so that everyone who's annotating can chat with other people who are annotating. When you get a vessel that's kind of frustrating or an answer you don't like, you can chat about it. And we think that'll be a great way to make our community a little bit tighter. So we hope you'll stick with us. We think there are exciting times to come. Chris, it's been a pleasure to have you on this. Thanks so much.
Thanks for inviting me. Yeah, this is fantastic.
Yeah, so we'll see you on Wednesday, hopefully, and keep catching. Thanks a lot.